Now this is a table of the different organelles and their functions that you're going to want to review. Um, you should already know them. There's nothing new there, so you do need to review through these. But we're going to talk about these organelles now. We're going to start with the nucleus, which is what you find in your eukaryotic cells. And it's surrounded by this nuclear envelope or nuclear membrane that is a double layer, and it's got these pores in it so that things can move in and out of the nucleus. It looks kind of like Swiss cheese. Inside this nucleus, we've got our chromosomes where our DNA is. And then the dark, dense area is called the nucleolus. And this is where we have ribosomes are made, and ribosomal RNA is also synthesized in this area. Now, we know cells have to divide to grow and develop. And this is just showing you mitosis, which is a nuclear division, where we start out up here with a cell that has a cell membrane, and you see the nucleus, the nucleolus. The cytoplasm's going to have all those organelles, that's just not drawn in pictures of mitosis. And those cells, or that cell, is going to go through the cell cycle. And so in interphase, this is not part of mitosis, but this is when the cell grows and it replicates its DNA, and it replicates all of its organelles. So you notice back here we only had 2 centrioles, and now we have 2 pair, and we've got double the amount of DNA, and the cell's bigger. Then it goes into mitosis, and it starts with your prophase, and I've lost my mouse, there it is, where that chromatin, the DNA, is going to thicken up and make these chromosomes. Our nucleus is going to disappear, the nuclear envelope disintegrates, and those centrioles are going to start moving to opposite sides of the cell, and the spindle fibers are going to form between them as it moves. In early metaphase, you can see how the centrioles are on opposite sides, and this football-shaped structure, the spindles, how the chromosomes are attaching to the spindles by their centromere, and they're beginning to line up in the middle, and by the time metaphase is finished, they are lined up in the middle. In anaphase, you notice the cells get longer. It's stretching out, and as it stretches, it pulls the sister chromatids apart, and they travel towards the opposite poles of the cell. And then when we go into telophase, you see that cytokinesis is beginning because the cleavage furrow is starting to pinch in, and eventually it'll pinch in all the way, producing 2 cells. The nucleus and nucleolus, nuclear membrane, are reforming, and the chromosomes will thin back out and make chromatin, so you're left with 2 new cells. So you might need to go back and review mitosis if you're not familiar with it. Now, looking at the endoplasmic reticulum, we have rough ER and smooth ER. And endoplasmic reticulum is a double membrane, and a lot of things move through this double membrane. So a lot of times we describe it as being for transportation. If it's rough ER, RER, it has ribosomes attached to it, and those ribosomes make proteins that are then moved into the ER where they're packaged and transported. If it's smooth ER, like you see down here in the bottom picture, it has no ribosomes, but it functions in the processing and storage of lipids that will move through it as well. The Golgi apparatus is where those proteins go after they're made and move through the ER. They're going to go to the Golgi apparatus, and the Golgi apparatus is just a stack of flat membranes, and like you see here, and those stacks are called cisterna, And those proteins are going to be modified and packaged and shipped off. So this is kind of like the mail room or the shipping department in a factory. So the way things are made and moved, ribosomes are made in the nucleus, and they move out through the rough ER, and they make proteins. And the proteins then move to the Golgi apparatus, where they are modified and packaged up into little vesicles that are then secreted by exocytosis from the cell. Now when we talk about energy, you know we talk about mighty mitochondria. This is what makes our energy, our ATP that our cells use. So if you got a cell that has to have a lot of energy, like a muscle cell, it's going to have a lot of mitochondria. Chloroplasts are only found in cells that are going to perform photosynthesis, because they convert the energy of the sun into chemical energy. And you find these in algae and plant cells, and this makes most of the organic nutrients for other organisms. Talked about ribosomes a little bit already. They're made of RNA and proteins, and they can be scattered around in the cytoplasm or on the rough ER. They're going to be larger than prokaryotic ribosomes, but they do the same thing. Protein synthesis, making proteins. Now the cytoskeleton gives some structure and support to the cytoplasm and the organelles that are in it. It's a network of proteins and microfilaments and microtubules scattered around in the cytoplasm, and that's what the organelles anchor to and the way they move throughout the cell. This chart is a comparison of your prokaryotic cells, your eukaryotic cells, and your viruses. And if it's got a plus, that means most members of that group have that characteristic. If it has a minus, most of them don't. But if you got the plus and the minus, that means some have it and some don't. So just looking here at the nucleic acids, DNA and RNA, all of them have them. A true nucleus is only in the eukaryotic cells, so the nucleoid region is going to be found only in the prokaryotic cells. And the only ones that go through mitosis are eukaryotes. And you can see they both prokaryote and eukaryote have ribosomes. You don't see it in viruses though. Viruses don't have a lot because remember, they're not living, they're just particles, and they have to have that host cell to survive. Continuing with this chart, you can see that our viruses are going to have capsids instead of a cell wall. And our eukaryotes, some of them are going to have a cell wall and some of them aren't. And look at the size. That's micrometers. So you can see your eukaryotes are the biggest, the viruses are the smallest. So our next concept check, the eukaryotic organelle that's responsible for transporting vesicles inside the cells is Golgi.